This video is about polarized optical microscopy, or POM for short. This microscopic technique makes it possible to see anisotropic domains within samples using visible light. My name is Cedric Genestra, and I'm a graduate student at Rice University. This video is made as part of the final project for ELEC 571. Today is May 5th, 2020. To start, let's review what visible light is. Visible light is a small part of the electromagnetic spectrum between infrared and ultraviolet light. Electromagnetic waves are characterized by their wavelength, which is the distance between two peaks on the wave. Visible light consists of electromagnetic waves with wavelengths roughly between 700 and 400 nanometers. Some really useful technologies make use of the whole range of the spectrum: communication, microwave ovens, night vision, sterilizing N95 masks, and medical imaging, just to start. For this video, we'll only look at how visible light is used in POM. Let's first break down an electromagnetic wave. It's made of two components, an oscillating electric field and an oscillating magnetic field. These oscillate in planes that are perpendicular to each other. So what is polarized light? Well, light can actually be polarized in two ways: linearly and circularly. To simplify the drawing, let's consider looking straight at the light wave as it propagates towards us, so we see the original axes from this perspective. Let's also only draw the electric component of the electromagnetic wave. If we were looking at a polarized light source with our eyes seeing lots of electromagnetic waves, all of the electric field components of each wave would be pointing along the same line. In our case, up and down. Light can be linearly polarized along any direction, so all we would have to do is rotate the light, or in this case, the page, to change the polarization direction. Circular polarization is a bit more complicated. Briefly, instead of the polarization direction remaining in one direction, the vector that describes the electric field rotates around a circle in a clockwise or counterclockwise direction. What's interesting about this type of polarization is that it does not change with rotation. As I rotate the page, the direction of polarization is still counterclockwise. Polarized light can be created from light sources that we deal with in everyday life, like the sun or incandescent light bulbs. Both of these are sources of unpolarized light. Remember, unpolarized light has a mix of photons with their electric fields oriented in every possible direction, indicated here by these arrows. So how can we make polarized light? One way to make linearly polarized light is to shine unpolarized light through a polarizer, which allows only the components of the electric field that are aligned with the polarizer to pass through. In this case, only in the vertical direction. In this process, some of the intensity of the light is lost according to cosine squared of the angle theta. For unpolarized light, the average angle theta for any given direction is pi over 4, or 45 degrees. This means that the intensity of the now polarized light is 1/2 of the original intensity. Now, what would happen if we added another polarizer in series, called an analyzer? Applying the same equation, we find that the intensity of light leaving the polarizer should equal 1/2 I naught cosine squared alpha, the angle of the analyzer with respect to the polarizer. Looking at this equation, we see two special cases. For alpha equal to 0, I2 equals I1. No intensity is lost. For alpha equals pi over 2, or 90 degrees, I2 equals 0. In other words, no light is transmitted through the analyzer. As we'll see, in POM, the polarizer and analyzer are kept perpendicular. This may seem odd at first, but light interacts with the sample such that we don't just see a dark image. To perform POM, you first need a light microscope. The light source emits unpolarized light, which is transmitted through a polarizer. Polarized light passes through the sample and is sent through a lens and analyzer before being recorded by the detector. If your sample is made of normal material, transmitted polarized light maintains the same direction of polarization. If your sample is made of birefringent material, polarization of the transmitted light is rotated. Because the transmitted light has some non-zero electric field components aligned with the analyzer, light is able to reach the detector. Birefringent materials have the property that the refractive index depends on the relative orientation of polarized light with the optical axis of the material. Polarized light travels at different speeds when traveling parallel or perpendicular to the optical axis. The delay between the slow and fast rays is called retardance. We can observe birefringence in materials that are anisotropic, meaning that some properties change depending on the direction. When studying birefringent materials using POM, we're identifying areas of the sample that have some structured molecular alignment. Typically, several images of the sample are taken with different orientations of polarization. This can be achieved by physically rotating the sample or the crossed polarizers. The images at different polarization angles will have different levels of illumination. An area that appears dark is either isotropic or a birefringent domain with its optical axis parallel to either the polarizer or the analyzer. A birefringent domain will appear brightest when its optical axis is at a 45-degree angle to both the polarizer and analyzer. Some examples of birefringent materials are stretched polymers, solid crystals, and liquid crystals. In my own research, I use POM to study solutions of carbon nanotubes. At high concentrations, carbon nanotubes align in small liquid crystalline domains. These solutions can be used to make high-strength conductive fibers. POM has also been used for clinical diagnostics of the progression of cancer. Cancerous cells produce lots of fibrous proteins, which birefringe when they align. For this type of application, POM is usually coupled with Muller matrix analysis, which mathematically describes how light interacts with the sample. Other optics can be added to provide quantitative information. Compensator plates increase the contrast of birefringence and are used to determine the value of retardance. In this video, we've seen that POM is used to study molecular order in anisotropic materials in qualitative or quantitative ways. POM is useful for a wide range of applications, from material science to medicine. Thanks for watching, and see you next time.